Okay, thank you all via Zoom or in person here for coming to this week's Kansas Biological Survey Center for Ecological Research weekly seminar. I'm Dan Reuman. I'm just here to introduce the speaker. This is John Walter. I'm very pleased to introduce John Walter. He did his undergrad at Gettysburg College partly in English. He's got a really interesting history. That's part of it. Then he went to do a PhD at UVA. He did a postdoc at VCU and then a postdoc here. And then he did some interesting transition. If you are a grad student who's thinking about alternative career pathways that still involve research, John knows all about that. He's done everything from fellowships to starting his own NGO in order to be able to support his research. And now he lives in Virginia, but he works at UC Davis. You ask him how that works. His interests are quantitative ecology, and he's transitioned gradually from a population focus to now working across scales and doing more ecosystem sciences work. So here he is, John Walter.
Yeah, thanks very much. It's a pleasure to be back in Lawrence after like 5 years since I was a postdoc here. And it's really a privilege to be able to travel and to actually like see and talk to people in person again. So thanks for making that possible and for coming to see this talk in person or virtually. So as Dan mentioned, I have kind of been transitioning my research career from one that when I, let's see, trying to talk and negotiate Zoom at the same time, and I'm kind of failing at both. And so the last time I gave a KBS Friday seminar in 2016, I talked about allele effects, which reflected at that time in my career a focus on population dynamics of insects, especially as governs. Actually, do we want to switch the lights so that this shows a little bit more clearly? And so, yeah, earlier in my career, I was very interested in insect population dynamics and how they manifest in invasions and in damaging outbreaks. And since
Move that thing for you.
Yeah, how do I get?
There you go.
All right. And since then, since coming to KU, I became more of a quantitative ecologist and started increasingly using statistics and mathematical modeling to understand a wider variety of study systems and across different organizational scales in ecology. And that's kind of led me to this new problem in my research, understanding why lake ecosystem trajectories, temporal patterns of these systems are so heterogeneous, and really in spite of global change drivers that would lead you to perhaps think that many lakes would be changing in very similar ways. So before I begin, I'd like to just acknowledge some collaborators on this work, including some folks that are local to KU, including Jude Castens, who I began collaborating with when I was here, and kind of continued to pull him into a project or two. And Nat Coombs, now a PhD student in Dan's lab, who as an undergraduate conducted research with me. And I'll talk towards the end of this presentation about some of the work that we did together. This is also kind of a fun talk for me because normally I begin talks by trying to explain some complicated concepts that I've kind of worked on some theory or some methods for. But this time I get to start talking about why the study system I'm looking at is important. So kind of, yeah, moving back in that direction. So lakes are quite important, and quite important globally. They're an important source of drinking water and irrigation, and source of power. They provide recreation and food for many people, and they're an important component of carbon and nutrient cycling, especially because a lot of what we do on land ends up in our waterways. One of the major threats to water quality are algal blooms, and these aquatic wildlife, they can sometimes produce toxins depending on the composition of the species that make up that bloom, and those toxins can be harmful for wildlife, for people as well, and of course reducing the kind of perceived and recreational value of these systems. And we know that these are caused primarily by excess nutrients. And this has, in part, in concert with climate change, helped to contribute to a rather pervasive notion that blooms are, in general, getting worse. So becoming more severe or more frequent. And this is in part because nutrient consumption has increased many-fold over time, leading to higher nutrient concentrations that in waterways that can support these blooms. And also some important impacts of climate change. So warmer temperatures can favor blooms, especially of cyanobacteria, which produce those toxins that I was mentioning earlier. And it's not just temperature. Changes in the volume and the timing and the intensity of precipitation can alter the delivery of nutrients to water bodies in ways that may favor blooms. And so in some of my recent work, which was led by my colleague Grace Wilkinson and came out earlier this year, we decided to apply some long-term monitoring time series to this question of, well, are blooms really increasing in a widespread way across the landscape? And so we pulled together data from the Lagos program, which is a very valuable program to synthesize data sets on a large number of lakes in the US, and analyzed long-term trends on 323 lakes in the Northeast and the Midwest. And you see the locations of those lakes in the maps on the left. And the color indicates whether the long-term trend in the concentration of chlorophyll A during the summertime, when most blooms occur, is positive. So lake is getting greener, those are in green. If it is negative, that is colored in blue. So you can kind of see that there's a wide variety of these trends. And then on the right here, we have a histogram showing the regression coefficients describing those trends. And one of the things you'll notice is that it is centered very close to zero. So a similar number of lakes decreasing as increasing. And these percentages in the upper corners of that figure indicate the percentage of those trends that were statistically significant. And so we have about 10% of lakes, according to this analysis, becoming significantly worse. A relatively similar number actually exhibiting significant declining trends in bloom indicators. So this was for annual mean chlorophyll A. We looked at a couple other attributes of seasonal patterns of chlorophyll A, and they kind of all told a similar story to this. So this is a little bit of a contrarian take, and this there were some mixed reactions from the community about this work, and so I want to talk a moment and mention some things that we kind of do and don't mean. So for one, we don't mean that specific lakes aren't experiencing worse or more frequent blooms. Of course, we do see that a number of lakes are. And so some of the reactions we've gotten, well, kind of like, well, this lake that I've studied for years has been, getting worse. And so we're really talking about, synthesizing over many lakes and trying to understand the general commonness of this phenomenon and how it differs across lakes. We also don't really directly say anything about cyanotoxins. So our time series, we analyzed chlorophyll A. This is a kind of a bulk indicator of how much photosynthesis is going on, but it better captures phytoplankton, which do not produce toxins, than cyanobacteria. And because of the production of toxins by cyanobacteria, that's something that people are particularly concerned about. And one of the concerns, of course, is that warming temperatures lead to increasing dominance of cyanobacteria in lakes. And so we're not really able to say directly anything about that in this study. We focused on chlorophyll A because it's a widely collected indicator over long time spans in our data set, 10 to up to 40 years. And we can't do that same kind of analysis as broadly over the same longer timescales with cyanotoxins. But I heard recently Jude Castens mentioned some interesting work that Ted Harris is leading, looking at cyanotoxins in reservoir sediments as they accumulate. And that's a really interesting approach to me that really stands to help develop these longer term time series of cyanotoxins that might help to answer questions about those blooms specifically. So, but the thing that we do really want to take away from this analysis is that the trends that we're observing are quite heterogeneous among lakes. And I think that there's a real need, at least something that I'm very interested in, is trying to better understand why. So the kind of overarching question for the rest of my talk is trying to get at why temporal patterns of lakes are so heterogeneous. And in this paper, we took one crack at that by analyzing how that trend coefficient varies according to a variety of characteristics of lakes. And kind of what came out of that was that we do see some differences among regions in the mean trend coefficient for that region. We see that larger lakes may be more likely to be experiencing bloom increases. And we see this interesting interaction between the average chlorophyll A concentration for a lake and whether precipitation in the lake's watershed tends to be steady or declining or increasing over time. And it's kind of interesting to me that this interaction effect shows that in areas with increasing precipitation, and as we go towards greener lakes, we see a higher likelihood that that trend coefficient is going to become positive. And so one of the reasons I think that's interesting is because even though our overall conclusion is a little bit contrarian, that blooms are not increasing in a widespread manner, this relationship between precipitation and blooms does kind of point at some of the mechanisms by which we're concerned about climate change increasing blooms. So I've worked in Dan's lab and with Dan for a number of years now. And so you might be anticipating the pivot that I'm about to make from talking about trends to talking about spatial synchrony in aquatic ecosystems. And so when we talk about spatial synchrony, we're talking about this really widespread phenomenon in ecology where there's a tendency for fluctuations in species abundances, or in the case that I'll be talking about today, lake chemistry, for those fluctuations to be correlated across multiple systems. And so to illustrate that, you can just imagine, 5 lakes across Wisconsin. Of course, in the real world, they're not arranged in a perfect diagonal. But we could commonly think of lakes as sort of fluctuating independently from one another. If they were synchronous with one another, they would look more like this, where the temporal patterns, the peaks and troughs in these fluctuations tend to coincide through time, indicating that that relationship, that synchronous relationship across these systems. So a few additional things about spatial synchrony are that shared environmental fluctuations are a major mechanism of this phenomenon, tending to bring different systems in different places into sync with one another. And this is because things like climate and weather tend to be correlated over long distances. But if we observe synchrony, that means that both the driver that is acting on these systems and the way that the system is responding to those is similar. And in kind of population ecology, where I have my roots, we often can assume some amount of similarity in responses to environmental drivers. But as a bit of foreshadowing, maybe lakes are somewhat different. Another thing that I think is important to note here is that synchrony usually ignores these long-term trends, like I was talking about in the first part of my talk, in order to focus on these fluctuations and whether they are coincident in time. And this is something that is usually good for inference because spurious relationships and trends are more likely. Things are more likely it is more common for things to be trending in the same direction for unrelated reasons than for the fluctuations around those trends to be related to with one another. And so it can be good for inferring mechanistic relationships between things. And we, over a course of a large body of work in more so in population and community ecology, we know that controls on fluctuations are often also controls on trends. So one question you might now be asking is, how synchronous is Lake Chlorophyll A? So we took a subset of the lakes from that previous analysis that had longer time series, 20 or more years, and measured the synchrony between these lakes and their chlorophyll time series. And we did a thing that we often do, and we plotted this over distance. So in the blue here, you can see the typical, sort of the average pattern of how spatial synchrony in lakes declines with distance. We did the same thing, except we took growing season NDVI from the watersheds that the lakes are situated in, and measured spatial synchrony in that. And by comparison, we see a much greater degree of spatial synchrony on land, very high, near 0.9 or so at very short distances, and then declining, but still remaining positive over about 2,000 kilometers. But one of the things that's interesting about this is just looking at this relationship is a bit misleading. If we plot the raw correlation values for these lakes, we see this big scatter. We see that the rate, the scatter is still basically centered near zero, but we see that fairly large values, fairly large positive and negative values in lake synchrony are possible over basically all distances. So lakes can be synchronized with one another, but distance doesn't do a good job of describing which lakes are synchronous with which other lakes, which leads to the question of what does control chlorophyll A spatial synchrony. And so for this analysis, instead of taking a big data synthesis approach, I'm going to zoom in on a set of lakes, set of well-studied lakes. These are the North Temperate Lakes, LTER lakes in Wisconsin. And we know an awful lot about these lakes. They're extremely well studied. And these 7 are a couple of bog lakes, 3 seepage lakes, so lakes that don't have outlets and inlets, and then our drainage lakes that are connected to other water bodies through streams. And these are also arranged sort of loosely from small to large. And this is just a map of this area. So this matrix that you see shows the spatial synchrony measured as Spearman correlation between pairs of lakes. And I've kind of drawn these lines to separate out our bog lakes, our seepage lakes, and our drainage lakes. And you can see that the bog lakes are fairly synchronous with one another. The drainage lakes are fairly synchronous with one another. And our, as we go from our our smaller seepage lakes toward our larger seepage lakes, they become a little bit more correlated, a little bit more synchronous with the drainage lakes. So of course, this is a very small sample of lakes. So the strength of inference about those patterns is not strong, but we can kind of, I think, use this to start to generate some hypotheses about what's going on here. And so what seems to be the case from this set of lakes is that hydrologic connections and lake size, which is maybe not a mechanism, but is an attribute of lakes that's related to a lot of other things about them, play this role in structuring spatial synchrony among lakes. And this might look familiar if you're especially the sort of person that has thought a lot about spatio-temporal dynamics of aquatic ecosystems. So this analysis is sort of an update on a now 20-year-old analyses of these same lakes. And there's been some previous work that has also similarly concluded that lake size and lake hydrologic connections are important to structuring their temporal dynamics and their synchrony. But because of the time that has passed, we now have a lot more data that can help us to disentangle mechanisms, and that's particularly as the length of these time series has accumulated, and we have now data on many more lakes that can help us to understand generality, maybe have the statistical power to tease apart some of the hypotheses that I advanced about what's structuring spatial synchrony in those NCO lakes. We have methods now that are improvements over traditional ones for uncovering new patterns and strengthening inference about mechanisms, and some different conceptual frameworks that have grown alongside this method that can lead us to ask different questions that I think hopefully can be productive in trying to understand the heterogeneity in lake temporal dynamics and what really structures that. So I'm going to talk about a couple of studies. One is focused on temporal coherence between lake and landscape primary productivity, and then timescale specific synchronous fluctuations in the biogeochemistry of Adirondack lakes. And there's some jargon in there, and I'll hopefully clearly explain what those terms mean. So this first study on how related lakes are to their watersheds in terms of their primary productivity came out last year. And if you're interested in it, there's a citation. I'm going to talk just due to time constraints about just some highlights from that work. But please feel free to check that out and ask questions if it interests you. So one thing I want to start with is this terminology of coherence versus synchrony. And this is something that kind of inherits from some of the methodological tools that we've used this. But the way that I'm going to use these terms, and there are some differences in the literature, are that synchrony implies this kind of in-phase relationship between two variables, and in-phase being equivalent to a strong positive correlation. The peaks and the troughs of these fluctuating time series coincide with one another. Coherence, as I'm using it, generalizes to other kinds of persistent relationships between these variables that might be time lag. So there's a consistent offset between the peaks and the troughs of the time series, or being in anti-phase with one another, equivalent to a strong negative correlation where the peaks and the troughs are aligned but different in the two time series. So we actually, this is some of the same data from those distance decay figures that I showed a few slides ago. And we wanted to use that to ask, are lakes coherent with their watersheds? And why would we think to ask this question? It's kind of a strange question in some ways. Well, there's some ways that coherence might arise. So the lake and the landscape that's embedded in are experiencing similar climate and weather drivers. And so those, particularly if they act simultaneously but independently on the lake and its watershed, might tend to produce synchronous relationships in primary production between those two, in the kind of thing that you can only, is just so ludicrously reductionistic that you can only get from someone who is sort of a theoretician. Photosynthesis is photosynthesis, right? Like it needs the same basic ingredients, whether it's in the water or on land, and these weather patterns shape controls on that. But there are also, things that are happening on the landscape that are influencing aquatic ecosystems through, transport processes, through runoff, and that might be mediated by certain features of the landscape, like wetlands. So first, the simple question is, well, are lakes actually coherent with their watersheds? And the answer here is sometimes. So this is a statistic that its range is bounded by 0 to 1. And so we see, and many lakes having, or the most frequently observed amount of coherence is, sort of middle of the road there. But it's really interesting to me that we see this, basically values over the entire plausible range of this statistic. So this is another way that lakes are really heterogeneous. And can we start to explain some of this? So in order to do that, we through a whole bunch of attributes of lakes and their watersheds and the environment that is influencing them into a random forest, and sort of a kitchen sink, throw it all into a kind of blackish box and see what comes out as being a good statistical predictor of that. And so this is a plot of the top several variables ordered from greater random forest variable importance to less. And we see a couple of things that come out. The atmospheric deposition of nitrogen tends, lakes or areas that have more deposition tend to be less coherent with their landscapes. And to be honest with you, I don't have a good explanation for why that is. An instance where I have a clearer idea of what's going on, or what could be going on, is that in places where the NDVI, the index of productivity of that watershed, the terrestrial component, where that time series is more variable, there tends to be greater lake-land coherence. And there could be a couple of reasons for that. One is maybe both are variable because the environment is more variable. And so there's a stronger influence of environmental conditions, namely climate and weather, on the fluctuations in both of these systems. And that tends to bring them closer into alignment with one another. Another possibility, if we're thinking about carbon that is fixed on land becoming a subsidy to aquatic ecosystems, maybe that larger variation in NDVI kind of provides a stronger signal, a stronger force on the lake, and helps bring it into coherence. But the other thing that we see a lot of is variables related to wetlands and runoff, which suggests this important role of the kind of hydrologic connections again in shaping the variability and the strength of this coherence. So kind of sum this up, it's plausible, but it's widely varying. And it seems to be related both to external drivers and to mechanisms related to transport of nutrients and organic matter kind of across the interface of these systems. So the next study I'll talk about is something that Nat contributed to when we worked together while he was a student at UVA. This work is unpublished. There are some things that we're still trying to make sense of. And so be happy to hear your feedback and comments and questions about this. In this study, we're looking at timescale specific synchronous fluctuations in Adirondack Lakes biogeochemistry. So first, what do we mean by timescale specific? So environmental time series are often composed of content that reflect different processes that are superimposed on one another. And these processes can have kind of different characteristic timescales of variation. So it's, in really, high resolution time series, you can see things like diurnal and seasonal patterns, but there are also climate oscillations that manifest over interannual timescales that can that have their own characteristic timescales of variability. And what that means is that the variation on particular timescales can be driven by different processes. And traditional statistical approaches like correlation don't do very well with this. And so, as probably many folks in the audience are familiar with, a big part of the research in Dan's research group has been developing and applying and trying to popularize methods that deal appropriately with this timescale specific variability and can tell us a lot more about what's happening and what are the mechanistic relationships that that produce it. The focus here is on lakes in the Adirondacks region of New York State. So upstate New York, this is an area that has been undergoing long-term recovery from acidification. And we've taken a nice monthly time series of data from 50 lakes. In this case, the chlorophyll A measurements are not particularly strong. So we're going to switch gears to thinking about dissolved organic carbon. And some of the reasons that we focused on this component in particular is that it has interesting and complex effects on aquatic ecosystems. There's sort of a nonlinear relationship between DOC concentrations and productivity in lakes. At sort of low levels of DOC, additions of DOC tend to bring with it nutrients that can stimulate productivity, but DOC also contributes to browning of waters, how colored the lake is, and so browner lakes, adding more DOC can create light inhibition that actually drives down productivity. Another reason this is kind of interesting to think about is that it's related to bioavailability of aluminum and its influence on fish. In this ecosystem, DOC is mostly terrestrially sourced. The systems that we're studying are pretty low productivity, and so a lot of what is entering our lakes is fixed in the forests and then is transported through hydrologic pathways into the lakes. And another piece of context is that because of this recovery from acidification, there has been a lot of work on the long-term trends in DOC and other lake chemistry properties. But there's a lot of variability around those trends. Those long-term linear relationships only explain about 10 to 20% of variability in DOC. So there's a lot that we can potentially uncover about what is shaping these dynamics. So we focused on coherent fluctuations with DOC that are shared across these 50 lakes. And so coherence relationships here imply that that there is spatial synchrony across these lakes. Again, we focused on DOC, but we quantified relationships with a number of attributes of lake chemistry and some external drivers that might be shaping its delivery and degradation within the system. So on the left, we have a bunch of negatively charged stuff. On the right, we have a bunch of positively charged stuff. Up top, we have some deposition variables, precipitation and temperature. On the bottom, we have some radiation variables, photosynthetically active radiation and UV radiation. These are important for photodegradation of DOC. So we've kind of mapped these relationships between all of these variables in DOC. The width of the links here indicate the strength of the relationship, with smaller p-values having bigger, having wider links drawn to them. And then there's the color codes indicate whether we're talking about an in-phase relationship, so positive correlation, an anti-phase relationship, negative correlation, and then stuff that's time lagged. And with the time lag variables, the arrowheads point toward the lagging variable. So for the relationship between precipitation and DOC, the arrow points to DOC. Peaks in DOC follow peaks in precipitation. And what you're seeing here is variation on approximately annual timescales. So we're looking at kind of the natural progress of the seasons in the system. And we can contrast that against what we see for interannual timescales. So dynamics that are playing out over 2 or more years. The two figures look really quite different with between each other. We see dropping of certain links, indicating that there is no longer a strong relationship between these variables, for example, precipitation and DOC, depending on which time scale of variability that we consider. And we also see some changing phase relationships. So these aluminum constituents that are related to fish toxicity change from a phase lagged relationship on annual timescales to one that is in phase on interannual timescales. So we are kind of still working our way through trying to understand some of the mechanisms that may be involved here. Certainly we see things like effects of precipitation, deposition of sulfate and nitrogen that can, in the case of precipitation, are sort of flushing DOC from the land into the lakes. And these acid deposition components are related to the breakdown of DOC within the lakes, as exemplified by those negative or anti-phase relationships. But I am cutting my teeth on biogeochemistry at this point, and still trying to better understand some of the interrelationships among the different chemical components of this system. Are they, responding to the same external drivers, or how important is kind of internal biogeochemical processing of these chemical elements within the lakes? So that's sort of an open question that I hope to be able to answer in the future. But I think we can kind of say in general, in a summative way, that these synchronous DOC fluctuations do reflect both external drivers and internal processing. And importantly, that these coherent relationships do differ across timescales, hinting at the timescale specificity of at least certain mechanisms in this system. So just to wrap things up here, we see quite wide variation among lakes and their temporal dynamics. And this reflects a combination of physical and ecological processes. There does seem to be a real importance of hydrologic connections in shaping the inputs and outputs into lakes, and that then influencing their dynamics. And one thing that I think is important is that we have a lot of pieces. We've studied a lot of lakes for a lot of time, but a lot of these studies have been on a single lake at a time or a small number of lakes, and there are really new opportunities, some of which that are partly exemplified by the work that I shared today, to gather data now on many lakes through this synthesis of existing data sets. Remote sensing of small water bodies is improving, and that offers other opportunities to get really broad coverage and longer term data on these systems. And we need this kind of work so that we can develop sort of greater certainty in what processes, what mechanisms, what patterns we expect across different systems generally, and the forces that are really shaping the context dependence and the wide heterogeneity that we see in these systems. So I will wrap there. Thank you very much for coming and for listening. And I'd be happy to take questions. I guess, put them in the chat box if you're on Zoom, or raise your hand if you're in person. Look forward to chatting. Matt.
So when you were looking at those variables for the synchrony of the different lakes in the larger data set, as part of the Rennet Forest model, or Rennet Forest evaluation of forests, or whatever the formal term is, along with runoff, did you look at presence of pervious or negative pervious surfaces around them?
So we looked at the amount of impervious surface in the watershed as part of that analysis, and that variable in particular did not rise toward the top, but I hope that I was being cautious in my interpretation, because it's a kitchen sink approach that is prone to overfitting on small variations that might not be mechanistically related to the variable that happens to pop up. So that was really kind of an exploratory analysis that hopefully will lead to some clear ideas that we can test, perhaps with data, perhaps with some additional experimentation that would lead toward a kind of more rigorous test of some of those mechanisms. Vadim, are you hand up?
I also like that. Result, not just kind of as a reminder that global change stuff is fun and makes things nice and easy, but it's actually typically it explains small portions. This is what you ignore the local things. But it was kind of really interesting to see that half of the trends are negative and half of them are positive. Right? So it's like that. Yeah, we do like conservation, and try to do like Do you have a sense of like what's driving that type of negative trends? Is it kind of nutrient management type stuff, or is it just the other half of changing climate?
Yeah, so one of the things that that we kind of thought about and waved our hands at a bit in that study was that we're looking predominantly at a period since the Clean Water Act, and that may be having some positive effects on I mean, certainly hopefully it's having some some significant positive effects on the health of our water bodies. That that's a national policy, so it doesn't necessarily in and of itself explain the heterogeneity in the patterns that we find. So we might need to look more towards local management, and we hope to be able to do that. We may need to get lucky with some funding to to put those data sets together and do the analyses. But yeah, that's something on our radar that I think is interesting.
Do you think it's less likely that environment?
I don't. So I don't actually have a good sense of the relative importance of environmental patterns versus environmental change versus management effects. Hopefully, we will be able to consider both together and resolve that. Yeah.
So I guess I would argue that management probably not changed that much in the last 30 years, but I might look more at whether or not there's
Yes, that is a good point. Yeah, so the invasive zebra mussels are definitely responsible for improved water clarity in some areas. And yeah, if we get this grant, we're gonna put a database of aquatic invasive species together as well. Yeah, I mean, these are just really complicated systems where there's a lot of different potential drivers going on. But I think it's an exciting time because we stand to be able to compile the kind of data that can help to tease apart some of those questions in a way that wasn't as feasible historically. Yeah, good point. Thank you. I think there's one in the chat. I'm gonna try and get to that. Okay, so the question in the chat is, how different is your approach from decision learning tree method? Also, I think AVHR spatial resolution is quite large. Yes, so the AVHR does have a pretty coarse spatial resolution. And we favored that data from that sensor because it's the longest term that was really available for quantifying that. So we wanted to be able to go back 20 or more years from the present. And so, things like, MODIS that have higher spatial resolution, kind of comparable temporal resolution and return times. But the time series just weren't quite long enough for that. Back to the audience, I think you had a question, and I'm sorry.
Just a general comment that maybe what it says is that the concept of lakes is sort of like the concept of invertebrates. it hides more than it really does. And that what you really got is a highly heterogeneous population of things with mostly standing water, and breaking that apart might be a more fruitful approach to understanding the process. Jude has some questions too.
Yes. Yeah, Jude, go for it.
All right. Thanks, John. Can you hear me?
Yes, I can hear you.
Excellent. Let me put my hand down first. I want to look like an idiot. Um, a couple very quick questions. Early in your talk, you mentioned, you used the word spurious trends are more likely in spatial synchrony. And I might know the answer. Does that mean it's easier to get false statistical significance with low frequency variations, or what do you mean spurious?
What I mean by that is if you're if two things are changing in general the same way through time, but are they can be mechanistically completely unrelated to one another, like the number of, I don't know, well, I won't try and think of a ridiculous example, but
I'll give you one.
yeah.
I mean, Jude and I both have a beard. Does that mean we're related? I mean, if you have a coarse description of something, then two things are more likely to be related according to that coarse description than if you have a detailed description.
Right, and so in that case, the more detailed description would be provided by the fluctuations around the trend than the fact that two things tend to be changing over the long haul in a similar direction. Does that make sense?
Yeah, no, that's pretty much what I was thinking. My favorite example is always the inverse relationship between global temperature and the number of pirates. Yeah.
That's exactly the kind of ludicrous relationship that I was not able to bring to mind.
The other question, so when you showed that dot plot early on too from that ecosystem study, and it was pretty wide band scattered points, there were still the width of the band persevered with distance, it seems, so that you had 1,000+ kilometers and still had pairs of synchronous samples. Is that real, or is that artifact or outlier luck or whatever?
I mean, the data are the data, so I'm going to say that the correlations are are real, and yeah, there are lakes that are quite far away from one another that appear to be fairly strongly synchronized, and lakes that are very near to one another that are negatively related to one another. And the magnitude of those positive or negative relationships are quite similar. So I think, yeah, I think distance does a kind of poor job of describing why that is, and hopefully I can help help start to resolve why there are those really long distance synchronous relationships, and whether there are mechanisms that actually explain that.
Yeah, I guess that was right. I mean, are there true teleconnections between these things? Are they or were those events just by the sheer number of points more extreme, like, again, kind of outliers or random occurrence, kind of like the large trends being related randomly?
Mm-hmm. Yeah, it's certainly possible that there are things that are related by chance for unclear mechanisms. I think the fact that we see that fairly consistently does suggest that there could be mechanisms that explain that, but what those are yet are not clear to me.
Last quick question, the Lagos data set is that, I know you can, well, you can download that. Has it been published online as a geospatial data set? And if not, can we do that?
It is, yeah, so it's published. Yeah, I'll chat with you over email about where it's located and all that good stuff. And that so the data that I analyzed was from a kind of precursor project that focused on the Midwest and Northeast. There have now been some data products released that are whole continental US, and a couple additional ones like the limnological water quality data are forthcoming. So
Very good. Thanks.
Yeah, so I have a question about that way early on in your presentation, you were just talking about the Northeast paper. Could you pull that up?
Sure.
Would it be too much of a pain?
no, it wouldn't be too much of a pain. I'll have to start sharing my screen again so that the folks on Zoom can see it.
So I just had a couple of questions about sort of the interpretation of them.
Sure.
Yeah, so particularly in the Northeast, was sampling just not done in like New Hampshire, Massachusetts, and Connecticut? Or are all the lakes that were sampled or non-significant for those?
Okay, so this is not just significant trends.
Sure.
These are lakes. So there are lakes. This is a compilation of predominantly state agency records, and so some of them are longer and better, quality and have more or fewer lakes than others. So there are, it's not like there are no sampled lakes in Pennsylvania or Massachusetts, as you said. But the lakes that were included in the data set didn't meet the requirements that we placed on them for the length of the time series and how many observations there were in each summer ice off season, so that we could get an estimate that we were comfortable with, that we were kind of capturing patterns that were meaningful. Blooms can be kind of temporally flashy, and so
I guess as a follow-up to that, if that's the case, what's going on in Rhode Island? Seems really homogeneous, right? And it seems like it's homogeneous in the direction that management would like it to be, right?
So that's actually getting greener. So that's
that's worse. Okay,
that's getting worse.
I see. All right. So why is Rhode Island so bad?
Yeah, I'm not sure. And that is that area, and also the Twin Cities are areas where we have pretty high data density, and there are a lot of lakes that are getting poorer. So it could be interesting to to kind of drill down into those data sets a bit more. But yeah, I don't have a good sense based on what we've done so far of why that is. I mean, there's definitely bias in how these monitoring programs are set up. This is not a completely representative sample of lakes in the US. That's why it might be interesting to try and do something like this with remotely sensed time series, where we can get a more spatially complete and less biased collection of data. But yeah, that's a good point.
So those ones in Rhode Island, they're getting more chlorophyll over time or less chlorophyll over time?
They're tending to get greener over time, more chlorophyll. Yeah, I mean, that's definitely a high population density area. I feel like that's an area that's been fairly high population density for a long time. And so there are certainly, land use and management changes that, that could and likely do influence the dynamics of these lakes. But I at least don't think that that's an area where we're seeing a lot of urban growth because it's been pretty dense for a long time.
I mean, you'd expect if that was an explanatory thing, that New York City and the region around New York City would be exactly the same.
Yeah.
I mean, I grew up in Rhode Island. I can give all kinds of reasons why I think that it's just like we pollute the shit out of it.
But it's getting greener. Right? It's the trend in Florida.
Getting greener is positive trend in chlorophyll.
Okay, so Rhode Island is negative trend.
Sorry, this is probably confusing. These two things. All right, this is something I need to fix for the next time I give this talk. Is the colors here on this figure are actually not related to the colors here. This is probably a source of confusion that has been pervading all of this conversation. So next time I give this talk, I will fix this so that the colors are consistent across those two plots.
I was really wondering why the Adirondack Lakes were getting worse.
Yeah, no, my bad there. Hey, this is why you give talks, is because you find out things that you mess up. Cool. There's, I think, one more question still in the chat that I hadn't gotten to. So looks like I failed to address part of a first question about the decision learning tree method. The random forests, they basically use like ensembles of classification and regression trees with random subsets of the data in order to sort of like iteratively test the fit of different combinations of variables. And so they are sort of an extension that's good for, kind of sort of data mining type approaches, trying to find your good predictors, especially when you have a lot of potential predictors. So they're very closely related approaches, but the random forest creates an ensemble of those. Thank you for the question, and sorry that I missed responding to it before.
All right, thanks, John.
Yeah, thanks everyone.